Hello everyone, welcome to my YouTube channel. This is Enoch's tutorial. Don't forget to like and subscribe to the channel after watching the video so that you can receive every update from this channel. In today's lesson, we are going to look at the structure and functions of biomolecules in the human body. We are going to look at proteins, carbohydrates, lipids, and nucleic acid. Today, I want my viewers to just watch the video and interact with me. We are just going to slow down or keep mute as we read the video or read the slides. If you have any question regarding anything here which you don't understand, just drop the question in the comment section so that I can address you there. So let's start by reading. Let's take the slide one by one and read them. So the first biomolecule we are going to discuss here is carbohydrate. And as been explained, carbohydrates are polyhydroxyaldehydes or ketones. So when you have an aldehyde group or an aldehyde compound with several hydroxy groups, that compound qualifies to be called a carbohydrate. We can group carbohydrate in several ways. So this shown here is the general formula for carbohydrate, which is CX then H2O. So as shown here, for every carbohydrate, it must follow this formula. So this formula gives a reason why we can also define carbohydrate as also hydrate of carbon, because it's just made of carbon molecules which has been hydrated. So they are called hydrated carbons. So based on subunit, we have several, or let's say three main categories of carbohydrates. We have monosaccharides, we have disaccharides, we have polysaccharides. Some books says monosaccharides, oligosaccharides, and polysaccharides. Yeah, same here. The first one here is monosaccharide, as shown here. They are just simplest carbohydrates, which is made of only one sugar molecule. So you have several examples of those. You have those that are made of 3 carbons, those made of 4 carbons, 5 carbons, and 6 carbons maximum. So examples have been shown in this slide or this paragraph. So you can just read them and understand. We equally have, so these are the examples of the monosaccharides. You have tetroses made of 4 carbons, you have trioses made of 3, you have pentoses made of 5, and hexoses made of 6. So listed here example of hexoses. We have pentoses, which are also shown here. Pentoses are mostly shown in RNA and nucleic acids, so you get it. So let's get the next slide. If you haven't read, finished, or if you are still on it, pause the video and read it. We equally have disaccharides, which are made of 2 monosaccharide units. Example can get sucrose, which is made of a fructose, which is a monosaccharide, and a glucose molecule. You can have maltose, which is made of 2 glucose molecules. Then you can equally have lactose, which is made of galactose and glucose molecule. These compounds or these sugars or carbohydrates are made of 2 monosaccharides, so we call them disaccharides. So the link that or the bond that links 2 monosaccharides to form disaccharides what we call O-glycosidic bond. So glycosidic bond is a linkage that links 2 monosaccharides to another monosaccharide to form a disaccharide. The name of that bond depends on the orientation or the type of carbons which are used in the bonding. If carbon 1 of one monosaccharide links to carbon 2 of another monosaccharide to form a disaccharide, we call it 1,2 glycosidic bond or 1,2 linkage as shown here. If it is carbon 1 of the first to carbon 4 of the next, this is what 1,4. If it is carbon 1 to 6, this is 1,6 glycosidic bond. Based on the orientation of the sugar in the aqueous solution, if the hydroxy group on the anomeric carbon, the anomeric carbon is the carbon which is having the double or the carbonyl group. If the carbonyl group or the hydroxy group on the carbonyl carbon or the anomeric carbon is shown down as seen here, it is called B or alpha glucose. If it is shown up, it is called beta glucose. So if the first linkage is from alpha glucose to, let's say, another one, then the glycosidic bond becomes alpha. If it is 1, it becomes alpha 1,2 glycosidic bond. If the first disaccharide or monosaccharide is an alpha sugar in which the hydroxy group on the anomeric carbon is shown down, it becomes alpha 1,4 or let's say 1,2 glycosidic. You can have alpha 1,4 glycosidic bond, alpha 1,2 glycosidic bond. You can have beta 1,4, beta 1,2, then also 1,6. So the alpha and the beta denote the orientation of the hydroxy group on the anomeric carbon of the first monosaccharide. If it is down, as shown in this case, it is alpha. If it is up, it is beta. So let's have also polysaccharides, which are made of several monomers of monosaccharides. So this one is a polymer of a monosaccharide. You can have starch, which is made of several residues of glucose, which are interlinked with alpha 1,4 glycosidic bond, as we explained earlier. So we have here several glucose monomers, which are interlinked with alpha 1,4 glycosidic bond, linear. So this is mostly the storage form of carbohydrate in plant. So plant stores sugars or carbohydrate as starch, whereas animals store carbohydrates as glycogen. So you have two forms of starch. You have amylose and amylopectin. So the amylose one is the one which is straight without any branches. So the linkage in amylose starch is just alpha 1,4 glycosidic link. But amylopectin is the one which has branches. So amylopectin, this one, is the one with the branches. So that one has two bonds. You have alpha 1,4 glycosidic bonds in there, then alpha 1,6 glycosidic bond. So this one is an amylopectin because it has alpha 1,6, then also alpha 1,4 in amylose. Amylopectin is just similar to glycogen because it also has the same bonding orientations. We have cellulose as another one polysaccharide, which is made also of glucose, but here it is beta 1,4. So the difference between cellulose and starch is beta 1,4 and alpha 1,4 in starch. It is the main component of plant cell wall. So this is just a summarized written note on cellulose. Take time and read it. We are going to have a soft question on the biomolecules. After this video, I'm going to put the link to some objective soft questions to biomolecules at the description section so they can have time and also read and see how best you understand the concepts of structure and functions of biomolecules. The next one is pectin. Pectin, which is also another polysaccharide. Let's go to the next slide. We have hemicellulose as another polysaccharide. We have glycogen, which we discussed earlier as another polysaccharide for the storage form of carbohydrates in animals. We have the chitin, which we discussed as the main component of the cell wall of fungi and also the exoskeletons of insects. So the components or the monomers are glucosamine. So that's for carbohydrates. Pause the video if you want to, or go back and watch or read about carbohydrate well so you understand the concept. Let's get to lipids. So lipids are basically a diverse group of chemicals that are either fat and oils. So the difference between fat and oil is that fats are basically solids at room temperature, whereas oils are liquids at room temperature. Whereas fats are from animals, oils are derived from plants. They burn in oxidation reactions to give several calories of energy as compared to carbohydrates. So with the same gram of lipids and carbohydrates, upon burning, the lipid yields more energy than that of carbohydrate. Carbohydrate is an immediate source of energy in the body because of its hydrated nature, which makes it more polar in the aqueous medium of the body. So lipids can be classified in several ways. You can have simple lipids like animal fat. You can have complex lipids like phospholipids and other compounds. So this is a typical simple lipid, which is just made of a glycerol backbone and three fatty acids to form what we call triacylglyceride. So an enzyme catalyzes this reaction, the reaction of lipid synthesis from glycerol and three fatty acids. So simple lipids are just made of a glycerol backbone and two or three. It can be one, two, or three fatty acids. So if it is three, it's called triacylglyceride. So it's called tag. If it is one, it is called monoacylglyceride, which is called mark. And if it is just two, it is diacylglyceride, which is that. So simple lipids are just made of two components, fatty acids and also glycerol. This reaction is more of a condensation reaction because it leads to loss of three molecules of water. So fatty acids are just like carboxylic acids, which are several residues of carbons. So we have several examples of fatty acids in the body. You can have myristic lauric acid, which is made of 12 carbons. You can have myristic acid, which is made of 14 carbons. You can have palmitic acid, which is made of 16 carbons. You can have stearic acid, which is made of 18 carbons, and arachidic acid, which is made of 20 carbons. We can have palmitoleic acid, which is made of 16 carbons with double bond, one double bond. You can have linoleic acid, which is made of 18 carbons with one double bond. Sorry, it is oleic acid rather. Oleic acid is made of 18 carbons with one double bond. Linoleic acid is made of 18 carbons with two double bond. And linolenic acid, which is made of 18 carbons and three double bonds. So they are just fatty acids which are made of several carbon atoms. Lipids in most cases exist as even number of molecules because they are synthesized in the body from a complex we call an acetyl-CoA molecule. Lipids are synthesized from acetyl-CoA, which is made of two carbon atoms. The acetyl group for extension is made of two carbons. So lipids are made of two or multiple of two number of carbons in most cases, unless under abnormal conditions. For complex lipids, we have several types. We have phospholipids. These ones are made of three or more different moieties in the molecule. So phospholipids are made of a glycerol backbone, fatty acids, and a phosphate group. for every phospholipid molecule, the phosphate group together with the glycerol form what we call the head or the polar head of the fatty acids, and also the non-polar end is that of the fatty acids. The acyl side chain, the acyl chain of the fatty acids is the non-polar tail. So because of lipids has both polar and non-polar end, they tend to form what we call a micelle in an aqueous medium, and they are described as we can have other examples of complex lipids. You can have gluten, waxes, you can have steroids. All these are examples of lipids which are very complex in nature. Now let's get to proteins. So we are done with carbohydrate, we're done with lipids. Now we are now going to proteins. The proteins are basically polymers of amino acids. So you can pause the video or go back and read on the ones we discussed and the lipids and carbohydrates so they can understand. As I said earlier, at the end of the video, I'm going to put a link to a video I did on the same channel on some questions on structure and functions of biomolecules. So you try your hands on and see how best you understand the concept. So amino acids are basically the monomers that forms a protein molecule. It is made of two functional groups. We call it an amino group and a carboxylic group. So there's an amino acid, it has a carboxylic group, it has an amino group, it has an alpha carbon that links the two functional groups, and a side chain. The side chain defines the properties of the amino acids. These two parameters gives a very neutral medium. So in some aqueous medium, the carboxylic acid will exist as a negative charge, whereas this one, the amino group, exists as a positive charge. So the net charge cancels out here. So the properties of the amino acid will be decided by the side chain of the amino acid or even the proteins, because they form the backbone or the protein molecule. So a typical example of amino acid, you have glycine. Glycine, that is if the side chain R group is H, it is glycine. If the side chain is methyl group, it is alanine. If the side chain is isobutyl group, it is called an isolate or leucine, as in here. We have several examples of amino acids. You have aspartic acid, you have glutamic acid, you have lysine, histidine, asparagine, and the rest. Proline, tryptophan, phenylalanine, tyrosine, serine, cysteine. These are the 20 known essential amino acids in the body. They are called essential amino acids because the body can't produce them. Therefore, they must be eaten in the foods that we eat. The ones that the body can produce them are called non-essential amino acids. So amino acids joins to form what we call a peptide, which undergoes polymerization to form a protein molecule. So peptides are made of one or two or more several amino acid residues joined by a bond we call a peptide bond. So a peptide bond is a bond that links carboxylic acid group of one amino acid to amino group of the next amino acid. The bond is called a peptide bond, or if you want to define from the terms or from level of organic chemistry, we call it an imide bond. This is an imide bond or a peptide bond, which is also formed from a dehydration reaction to form this linkage. Polypeptides, as the name implies, poly means that several molecules. So amino acids fold when they form a peptide, as seen here. If an amino acid forms a linear peptide, they fold. The sequence of the amino acid in the linear polypeptide or a protein is what we call the primary structure of protein. If the primary structure coils around to form helices or sheets, we call it secondary structure. Subsequent three-dimensional folding of the secondary structure forms what we call a tertiary structure. And two or more tertiary structures which are interbound forms what we call a quaternary structure. So you have four main levels of protein structures. You can have primary structures which are formed from the folding of just the bonding of the amino acid sequence, or the amino acid sequence in a peptide defines the primary structure. If the primary structure folds, you form a secondary structure. You form an alpha helix and a beta sheet. And also, if the secondary structure folds out to form a three-dimensional molecule, you form a tertiary structure. And the tertiary structure interfolding, or two or more tertiary structure interlinking to form a complex structure, forms what we call the quaternary structure of proteins. Now let's get to nucleic acids. So nucleic acids are basically RNAs and DNA. We have two main types of nucleic acids. We have RNA and DNA. RNA is ribonucleic acid. DNA is deoxyribonucleic acid. The name implies one is having a hydroxy group on carbon 1 or carbon 2 of the ribose sugar. The other does not. So RNA has, while the DNA does not. So we say it's deoxy. So there is no oxygen on carbon 2 of DNA ribose sugar. Nucleic acids are formed from monomers we call nucleotides. Nucleotides are made of three main components. We have a nitrogenous base, we have a ribose sugar, and a phosphate. Nucleotides undergoes a condensation reaction by forming what we call a phosphodiester bond to form what we call nucleic acids. When a nitrogenous base bonds with a ribose sugar, we form what we call a nucleoside. And when nucleoside combines with a phosphate group, you form a nucleotide. So you can have a mononucleotide, monophosphate nucleotide, a diphosphonucleotide, and triphosphonucleotide. When a nucleotide is degraded, as we said earlier, when it is degraded, it shows three main components: a phosphate group, a ribose sugar, and a nitrogenous base. Nitrogenous bases are of two kinds. We have one with two ring system, which we call purines, which are adenines and guanines, and the ones with just a one ring we call pyrimidines, which are uracil, cytosine, and thymine. Thymine and cytosine are found in DNA, whereas uracil replaces cytosine in RNA. So this is how a nucleic acid is formed. A nucleic acid is formed from DNA molecule is formed from two strands. Each strand is made of polymers of nucleotide. The backbone of each strand is made of the nitrogenous base and the phosphate. whereas the linkage is the backbone is just the sugar and the phosphate group, whereas the linkage is the nitrogenous bases. As seen here, DNA is a double helical structure made of two strands. Each strand is made of several monomers of nucleotide joined by a phosphodiester bond. So this linkage is made of several monomers of nucleotide, and this linkage is made of several monomers of the nucleotide. The nitrogenous bases from each strand joins through a hydrogen bonding and a complementarity system to form what we call the DNA molecule. Bear in mind, DNA is made of two strands. RNA is just made of only one strand. So if there's one strand here, thymine bonds with adenine, whereas guanine bonds with cytosine. Thymine bonds with adenine through a double bond a double hydrogen bond system. So two hydrogen bonds links thymine to adenine, whereas three hydrogen bonds links guanine to cytosine. So this is how basically nucleic acids are formed. They are formed by joining of two strands through hydrogen bonding and complementarity system. Per complementarity rule, adenine bonds with thymine and guanine bonds with cytosine. So it's a very short tutorial and a very short reading material on the structure and functions of biomolecules. Thanks for watching. Don't forget to subscribe to my channel after watching so that you can receive every update from this channel. Like so that YouTube can share this video to many people, and subscribe to. As I said, you are going to put a link to the description section just beneath this video, where you can get access to some solved objective questions on biomolecules, carbohydrates, proteins, and lipids, and also nucleic acids, so that you can test your understanding on how best you've understood this concept. See you in the next video. Bye bye.